today is we'll go over the work we've done so far on specialised cells with reference to sperm cells and egg cells. And as you can see, we're going to go through the work that you did on Monday. Then we're going to have a look at two types of epithelial cells, ciliated epithelial cells and epithelial cells with microvilli, and have a look at the task I want you to do with that. This is the last lesson where you're going to have some new input. So Thursday's lesson will be the last lesson before half term, where we'll be having a look at your model. So you've got to make sure that those models are ready for Wednesday evening, so you're in a position where you can review them and self-assess them on Thursday's lesson. Now, going through the work that you did on Monday, looking at the sperm and egg specialised cells. This is the sheet that I wanted everyone to do. The extension work I'll have a look at when I collect it all in, but we'll go through this because this is the real core information. So the first thing is looking at sperm cells. It states here that sperm cells are haploid, and what does this mean? Now, you might have had to look up this. We did talk through, and it is mentioned on the slides, but if a cell is haploid, the hap means it is half, so something about it is not complete. It only has half a set of something. Now, in this case, it has half a set of genetic information, so rather than 46 chromosomes, it only has 23 chromosomes. Now, that's really important because when a sperm cell fuses with an egg cell, we get two haploid cells fusing together to make a complete diploid cell, which means it's got a full set of genetic information. So the idea that half the genetic information comes from dad in the sperm, half the genetic information comes from mum in the egg, when they fuse together, there's a full set of genetic information. Next question. It states that the cells that make sperm cells are diploid. So then what type of cell division takes place and what must happen to make a haploid cell? Again, a little bit of independent research was probably needed for this question. To go from a diploid cell with a full set of genetic information into a haploid cell with half a set of genetic information, the cell will need to divide and split into two. Something that I've just noticed on my slide that I'll change for the sheet that you get, it says mitosis there. It isn't, it's meiosis. But I'll make sure that's correct when I upload the sheet for you. And the number of chromosomes will halve. So just to double check on that, I want to make sure everyone's very clear. The type of cell division is meiosis, and the number of chromosomes will halve. And what you can see on the screen at the moment is incorrect, but it will be correct on the document that you get in your assignment. Next, looking at the different types of cells. So sperm cells have several adaptations, and how are they adapted to suit the function of that cell? So first of all, points out here that the sperm cell has a tail. Now, the adaptation of the tail means the sperm can swim, so the sideways movement of the tail propels the sperm cell forwards. The sperm contains lots of mitochondria. This provides the energy needed for the sperm to be able to swim. And then the hard one, the acrosome. Now, the acrosome is an area on the top of, well, on the head of the sperm, and that contains lots of enzymes. Those enzymes can break through the jelly coat of the egg, so the sperm can fertilise the egg. Looking at the egg cell, so question 3, what is the function of the jelly coat around the egg? That is protection. It protects the egg, so it's ready and it's viable for fertilisation. How does the jelly coat change after the sperm cell fuses with it? The jelly coat surface changes, so no more sperm can get in, so the substances actually change to prevent the enzymes from working. What is the function of this jelly coat? In this case, is it prevents any more sperm from fertilising it, so you don't get more than one sperm fertilising the egg. That document with that meiosis correction will be in the assignment, so you can go through and check your own work against that. Now, today's lesson, I just started off by showing you this short little video and see if we can work out what's going on here. So if we just look at that. Now that's actually a video of inside the body, and you can see cells there, but you can see something on the cells. So that's what we're going to be having a look at today. So you can see there's some movement. Now, what's happening there is you can see something called cilia. Now, cilia are little finger-like projections that wave around and they move. We need to know what they are, where they are, and why they are found in those places. So, just breaking down what ciliated epithelial cells actually means. It's quite a complicated term. Cilia are the little hair-like projections that move, and I always tend to show these with my fingers and imagine them like moving fingers, but they're moving. Epithelial means a lining of any organ or tissue, so it might be the lining of the airways or the lining of the oviduct in the female reproductive system. So it's a lining or like a covering of an organ or part of an organ. So looking at the ciliated epithelial cell, you can see in this diagram here there are 4 cells, and on each cell there are lots of finger-like projections. Now those finger-like projections can move, they wave and they beat. The purpose of that is so anything that is being caught on those finger-like projections can be moved or swept along. So in this illustration you can see the egg cell is being moved along by those cilia. Now I've put a little question there, which is, why are there so many mitochondria in these cells? So if you just have a pause for a minute, pause the video, have a think about why there would be so many mitochondria in cells that have ciliated epithelial cells. Okay, going back through that, mitochondria are involved in respiration and they release energy for the cell. Why would these cells need lots of energy? Because the cilia are moving and that movement would require energy. Here's a microscopic image showing those cilia, so you can see long wavy hair-like projections. So breaking it down, what are they? They're cells with hair-like projections that move, that beat. Where are they found? Lining of airways and in the oviducts in the fallopian tubes. Why are they there? Well, if we look at the lining of the airways, in that image, if they were along the airways, they would be there to trap microbes that are caught in mucus produced by our airways. They beat and they move upwards and move that mucus and any dust or any microbes trapped in there into our mouth, and then we can swallow them and the stomach acid will kill them. Why are they in the oviducts or the fallopian tubes? The reason for that is shown in this picture here. It's to move the egg from the ovary further along the oviduct, so then if there's a sperm cell around, the sperm cell can successfully fertilise the egg cell. So ciliated epithelial cells are always about the movement of something. So in the oviduct, it's about the movement of the egg. In the airways, it's about the movement of mucus and dust away from the delicate lung tissue. So that's ciliated epithelial cells. A slightly different epithelial cell is an epithelial cell with microvilli. And if we have a look at these, the projections are smaller and they are not moving. So the function of these projections is to increase the surface area of a cell. Now, an increased surface area will mean that there is more area for substances to be absorbed into that cell. So you can see here, this is a cell found in the small intestine. So the function of the small intestine is to absorb nutrients. By making the cell have an increased surface area, by having lots of folded microvilli, it will make it more efficient at absorbing those nutrients. If the cells didn't have those microvilli, they would have less surface area and an insufficient or not enough nutrient would be absorbed into those cells.